Hello everyone. So the next topic which we will be covering today is about the cytokinesis. So what do you understand by the term cytokinesis? The term itself is depicting that cyto means cytoplasm and kinesis means division. So there is a division of cytoplasm. Fine. So in these slides, we will be covering all the details of the cytokinesis, its diagrammatic representation, and all the process in the detail in the plants and the animal cells. So let's get started with the first slide of cytokinesis. Introduction. So division of cytoplasm is called the cytokinesis. As I have told that cytokinesis means division of the cytoplasm. And cytokinesis accompanies every karyokinesis. So karyokinesis means there is a nuclear division. The mechanism of cytokinesis in plants and animal cell varies. So in both the cells the mechanism varies. So we will be discussing both these things in the separate slides itself. In animals it is achieved by cleavage, and in plant cell it is achieved by the cell plate formation. So in both the cells they have different processes and mechanism of cytokinesis. So what happens in the animal cells? In animals, cytokinesis is achieved when a contractile ring of the cell microtubules form a cleavage furrow that divides the cell membrane into the half. So what happens is the ring of the cell microtubules, they divide the cell membrane into two parts, into the half. So this process in the animal cell is attributed to the role of contractile rings, and they're held by the mitotic spindle. So there are spindle fibers which is attached by the mitotic spindles. So these microtubules and cell signals determine the location of the contractile rings, and therefore they direct the plane of the cell division known as the division plane. So this is known as the division plane, in which they direct the plane of the cell division. So if we look at the stages of mitosis, first there will be interphase, then prophase, then metaphase, then anaphase, telophase, and after the last process of mitosis is the cytokinesis. So that's why we are here discussing about the cytokinesis, because it is the last process of the mitosis. So this was all about the animal cell. So after this, the contractile rings which was formed, then the cleavage furrow they forms around the division plane, and even actually the pinches of the separating cell into two cells. So this is followed by the contraction and constriction of the contractile rings, which is made up of the actin, myosin, and the regulatory proteins. So these are the regulatory proteins which a contractile ring which is made up of. So what are the functions here? So actin and myosin, they are formed during the interphase. So they are formed during the interphase. Interphase is the first stage of the mitotic phase, which is assembled in the cortical network of the filaments filled with actin and myosin. As the cell division continues, the actin filaments are reorganized while the myosin accumulates during the anaphase to form the contractile rings. So what happens is these contractile rings, they are positioned by the action of the proteins such as actin, myosin, and regulatory proteins, and they act as a motor protein which allow the contraction of the muscle cells. So what happens in muscle cells? The muscle cells, they are packed, and they are packed with the actin filaments, the protein filaments, and they are pulled together by the myosin proteins. So here both the function of the protein has been depicted here, that it is packed by the actin filaments and it is pulled by the myosin protein, and they form an actin myosin ring which plays a role in the exclusion of the cytoplasm and the cell organelles. So they play a major role in the exclusion of the cytoplasm and the cell organelles. So here is the diagram which is depicting the cytokinesis part in the animal cell, that this part where these are the contractile rings of the microfilament, and this is the cleavage furrow, and on this there is a two daughter cells which are formed. So after the exclusion of the cytoplasm and the organelles, the ring and the microtubules are left behind and they form the mid-body structure which divides eventually. So this where the division takes place and converts into two daughter cells. So the cellular proteins, they cut and fusion of the plasma membrane are shut, while the extracellular elements, they held the cell together and they get dissolved the separating cells. So these are the two separating cells. So what happens is separated cell, they remain associated with the linked cytoplasm at the bridge known as the gap junctions. So they remain attached to the cytoplasm by a bridge known as gap junctions, which are formed by the remains of endoplasmic reticulum trapped in the mid-body structures. So we have introduced all the terms in the animal cells. What are gap junctions, what are mid-body proteins, what are contractile rings, what is it made up of, what is the role of actin and myosin proteins in the muscle, and what are how the contractile rings are being made, what is cleavage furrow. So we have all described the cytokinesis, that is the division of the cytoplasm in the animal cell. Next, we will be covering how the cytokinesis process takes place in the plants. So, what is the major difference between the plant cell and the animal cell? The major difference is that plants are made up of extra rigid cell wall, because the plants have cell walls and cell membrane, while the animals don't have the cell wall. They only have the cell membrane. So the plants are made up of rigid cell wall, and a special kind of microtubules are involved in the completion of the cytokinesis, which is known as phragmogoplasts. So this is known as phragmogoplasts. So phragmogoplasts, they are vascular spindle microtubules which is formed by the Golgi vesicles. Golgi vesicles is the cell organelle which during the telophase on the metaphase plate, they carry vesicles and cellular elements which cause the cellulose to the new cell wall. So this is how a phragmoplasts work in the case of plant cells. So these Golgi vesicles, which means these Golgi vesicles or the cell organelles, fuse at the center of the cell wall and they form the cell plate at the site of plant cytokinesis. So they play a role in the plant cytokinesis by forming the cell plate. So as the vesicle fuse, the cell plate continues to enlarge. They will fuse and the cells will continue to enlarge. So the phragmoplasts, they carry vesicles of the cell wall to the newly formed cell plate. The accumulated enzymes and the structural proteins and the glucose molecules between these membranes, which is formed by the Golgi apparatus, during the interphase, they contribute to the formation of the new cell wall. So they contribute to the new cell wall. So while the Golgi apparatus, they are incorporated and form part of the plasma membrane, and they form the part of the plasma membrane. So the cellulose which is carried by the pharmacoplast, they interact and combine, forming the complex and the strong rigid matrix of the plant cell wall, and they form the strong rigid matrix of the plant cell wall. So this is a diagram which is depicting all the stages of cytokinesis in the plant cell. So after the cell plate has been formed and divides the cell, the plasma membrane seals off, separating the two newly formed daughter cells. In between the two cells, there is a trapped endoplasmic reticulum which forms the plasmodesmata space and a gap which allows the passage of molecules from the one cell to another and helps in the communication of the cell. So this is the diagram which is showing the representation of cytokinesis. This is the anaphase. As I have told, there the mitosis occurs in 4 stages. The main 4 stages are prophase, anaphase, telophase, and the last is the cytokinesis. So what happens is there are spindle fibers which are attached to the chromosomes here, and Then after this, in the telophase, what happens? The vesicles is filled with cell wall components. So this is the green dotted things are the vesicles which are filled with the cell wall components. And in the next stage, that in the late telophase stage, these fuse to form a cell plate. And this is how a cytokinesis process takes place, a mechanism takes place in the plants, and there will be a newly two daughter cells are formed in this particular diagram. So this is how a plant cell of cytokinesis works. So these are all the references from which I have taken the diagram.